So we move on to the next talk now. We have two speakers, Colin Lee and David Posner. Both of them are, Colin is an MD PhD student, and David is a PhD student, both working at Professor Mena Clackworthy's lab University of Cambridge, talking about their beautiful paper, again, and their title, the talk is titled Humoral Immunity at the Brain Borders in Homeostasis. So take it away, guys.
Great. Can you see that? Excellent. Thank you so much.
Yeah.
Thank you so much to the organizers. Thanks, Rajan, for opening up with an amazing talk on meningeal macrophages. Right. So as was just mentioned, my name is David Posner. I'm a PhD student in Professor Mena Clackworthy's lab at the Department of Medicine in Cambridge, and this is a kind of beautiful image of our building. It's not quite Marseille, but it'll do. We're on Twitter, so please, find us if you have any questions or want to get in touch about any kind of exciting new science that you want to share. Right, so I'll just jump right in. So kind of a brief introduction to humoral immunity. I know there's a kind of a mixed audience, but B cells are an adaptive immune cell type, and they express this surface B cell receptor, which is an antigen receptor. Upon stimulation, these can develop and kind of further mature into a functional type called a plasma cell, which has the ability to secrete this B cell receptor into a solubilized form, which is called an antibody, and this is quite popular. And they come in various flavors, from kind of IgM to IgE. And not only do the flavors determine what the functions are, but they also determine kind of what tissue in the body they're prevalent in. And for example, IgA is classically found in the gut in great abundance, whereas IgG is found more systemically and is important in viral responses. But B cells and plasma cells also have other roles that are not just secreting antibodies. They also have functions in antigen presentation, for example. So B cells can capture antigen with their B cell receptor and then present it to the CD4 T cells via MHC class II. But they also have the capacity to produce cytokines and mediate or regulate immune responses, both in a pro-inflammatory and an anti-inflammatory context. And they can do this with various flavors of cytokines like IL-6 and IL-10 respectively. And both B cells and plasma cells have the ability to do this. And now on to the Dura. I don't have to go into huge detail because Rajan did an amazing job at introducing with some beautiful pictures. But just quickly, we're looking at a cross-section of a brain with a skull. And so here we got the brain at the bottom and the skull at the top. And you'll notice these little pockets of bone marrow in the skull. But we're interested in the Dural meninges, which are kind of here sandwiched in yellow. And what you'll look at is that we have these networks of big meningeal or dural vessels called venous sinuses. And the Dura is a really kind of special place when it comes to the immune interface and which antigens they're seeing. So they see antigens that they can experience through kind of the extracranial space, through the systemic circulation, so things like an IV challenge, but also they're experiencing antigens from the CNS parenchyma that's flowing through the CSF and then going into the Dura and getting picked up by meningeal macrophages, for example, and other cells. And we review that here if you're interested in checking that out. But basically, I'm going to focus for the kind of talk. We published a paper or a review article on humoral immunity. And so we mostly kind of focused on what's your phenotype and localization in the Dura and homeostasis, and what factors can influence the humoral immune compartment, for example, the age and the microbiome, but also what's their functional relevance, like why are they important? And we started with kind of localization, and we mentioned this in the article. So B cells, like all other immune cells in the meninges, love hanging around these big dural venous sinuses. And so what I showed you here, this major sinus called the superior sagittal sinus, but also the transverse sinuses, you're looking at it kind of in this orientation. And B cells, like T cells and other myeloid cells, hang around these big sinuses because they have access to antigen from the circulation, but also from the CSF that's being drained. And a really cool paper that came out of our lab in 2020 by an amazing student, Zach Fitzpatrick, looked at the kind of characterization of B cells and IgA plasma cells in the meninges. And what we can do is, if you take a kind of whole mount image of mouse meninges and you track every single B cell or plasma cell, you can then put it onto this kind of plot called a histocytometry plot. And so here we're highlighting this area is kind of above the olfactory bulb, and this area is towards the cerebellum. You can really notice that B cells and plasma cells alike hang around these sinuses. So this is the superior sagittal again, and the transverse sinus, showing that they're really kind of localized around these big vessels. And B cells are really kind of one of the predominant cell types in the Dura at homeostasis, making all, from 20 to 30% by flow, but it's also recapitulated by single cell. But other groups, like this paper from Simone Brioche from the Kelowna lab in the US, did an amazing job at characterizing by single cell, but also microscopy and flow, the kind of B cell heterogeneity that you can really see in the meninges in steady state. And not only did he find that there's kind of regular B cells shown by MHC class 2 and CD79B and CD20, but also found kind of the compartment includes developing B cells, and that's shown by RAG1 and CD93. And this is some beautiful microscopy from a RAG1 GFP mouse showing the developing cells along the vessels of the meninges, which is really cool. And so now that we know kind of the phenotype and localization, we can then go on to think about what factors influence both of these bits. And so we're really interested in kind of the microbiome in a way, and Zach did some amazing work on this. And so as I mentioned, IgA is the main type in the gut. And that was shared in the meninges. And so in the meninges, IgA plasma cells are the main plasma cell subtype. You also get some IgM and IgG, but mostly IgA. And that's been characterized by Zach really nicely. And what he also did was think about, what's the actual link between the gut and the meninges with regards to the humoral immune compartment. And what you're looking at here is a set of dandelion plots, which are a bioinformatics tool that was also developed from the lab from an amazing postdoc who is now starting his own group, Kelvin. And what he's analyzing here is B cell receptors in both B cells and plasma cells from the small intestine here in blue, and the meninges here in red. And it's color coded based on the subtype of B cell or plasma cell. So IgA we know is the dominant type in the small intestine, but you can also see some IgG and so on. But in the meninges, you see also predominantly these red clones, and they're spread, showing that there's kind of clonal expansion there. You also kind of see some IgG, but mostly IgA. And then you can also look at, if you focus only on the IgA compartment, you can see that these two organs share kind of about 20% of the clones in steady state. And what you're looking at here on the right two panels is every single BCR that's unique or shared across the tissue. And so in the bottom right here, you can see that all the BCRs that are unique to the small intestine are in gray, and all the ones that are colored are shared across meninges and small intestine. And you can see in the meninges, conversely, that pretty much most of them are shared or colored, showing there's a real relationship in the humoral immune compartment between these two organs, which is really, really cool. And you can go a step further from this, and Zach did, and we can look at a germ-free mouse. And so a germ-free mouse has kind of a depleted microbiome or doesn't have one since the beginning. And that reduces the humoral compartment, so the IgA plasma cell compartment in the gut, and similarly does so in the meninges. You can then go ahead and reconstitute, so you can return the gut microbiome, and then all the IgA plasma cells come back, and they come back in the Dura as well, showing that real dependence. But you can also play around with this and enrich your microbiome in one type of bacterial or fungal population. And here we enrich it with Bacteroides, which is a bit more inflammatory. And so we see that the IgA plasma cell the plasma cells come back, but they also come back with IgG plasma cells. And that is mirrored in the dural meninges, which is really cool. However, if you try to reconstitute just the skin, So you bring back the skin microbiome, that doesn't bring back the IgA plasma cells in neither compartment, showing this is kind of a gut-specific kind of axis. And then finally, why do we care about the humoral immune compartment? What is the functional importance? And this can be kind of split into three, or I've split into 3 slides. First, we're looking at, so a lot of the kind of meninges field has focused on autoreactivity with regards to the CNS, so kind of autoimmune diseases like EAE and so forth. But and so here we work from 3 groups or from multiple groups, but specifically these 3 papers were key in dissecting out the where these B cells were coming from. And so they found that in these pockets of skull bone marrow in the skull actually can give rise from stem cell to all these B cell precursors. And then these B cell precursors can enter the meningeal kind of layer or tissue via multiple signals like IL-7 and these like CXCL12, CXCR4 axis through these like really cool small channels that are both dependent but independent of vasculature. And those papers do an amazing job at kind of looking at by microscopy and some other models. But why are they going into the dura? And the answer is that they're potentially, and there's some really strong evidence for this, that they're being kind of filtered or against CNS antigens. So you want to get rid of all your CNS reactive B cells. And so if you're developing B cell here in the bone marrow and you enter and you autoreactive against MOG, for example, you want to get rid of that. And so this is the space to do so. And so these cells will be negatively selected and die, whereas cells that are not autoreactive against CNS antigens can go on and survive. So that's function number 1. Function number 2 was something that was also worked out by Zach, and that's really kind of protection of the central nervous system, so the brain, from circulating pathogens or challenges. And so here we got a kind of beautiful IgA plasma cell that has the ability to secrete IgA antibodies. And in the case of a Candida albicans infection, for example, that is coming through the circulation, these plasma cells are kind of the security that prevents this Candida from going into the brain and causing kind of a more fatal disease. And you can eliminate these cells with this treatment in red. So if you delete plasma cells and give a Candida challenge, these mice all die in this cohort within 14 days, showing that they're really, really important in kind of protecting from circulating pathogens. So that's function number 2. And then function number 3 is still kind of, I think, at its infancy, and it's really something that the lab is kind of new in, but really expanding, and this is a really cool field. And so as I mentioned, B cells and plasma cells can secrete things like IL-10 and pro-inflammatory cytokines, but through this kind of anti-inflammatory axis, they're able to control the composition of the dural immune system. And so in a paper from a PhD student, Marian Linnell, and a colleague, Stacy Krygar, in this paper, they kind of looked at the B cell compartment really closely, and they looked at behavior, and they saw that in a CD19 knockout at baseline, these mice have, so these are mice that don't have any B cells genetically. These mice at baseline have more anxiety-like behavior, or they exhibit these behaviors compared to wild type, showing that there's perhaps an axis that's dependent on B cells and plasma cells and maybe cytokines in kind of regulating behavior and cognition development. And in this CD19 knockout, they saw that the innate immune system and neutrophils are dysregulated. And so maybe B cells are controlling kind of, in a way, the immune compartment in the dura with these cell types. Finally, I'd like to thank loads of people in the lab, especially Zach Fitzpatrick, who kind of pioneered all this work, and of course my boss, Matt Clatworthy, who's a fantastic mentor and a great scientist. And then I'll kind of pass the baton to Colin, who's a PhD student in the lab as well, who has created this amazing resource for the field, and we'll kind of take it away from there. Thank you very much.